My name is David Oppenheimer, and I work in the Department of Biology at the University of Florida. Purchasing pre-poured polyacrylamide gels for routine protein electrophoresis is convenient because you can always have gels on hand that are ready for use. However, commercial gels can be expensive if your lab runs a lot of gels. Also, commercial gels are typically poured in plastic cassettes that are designed to be discarded after use, thus generating plastic waste that usually ends up in a landfill. Today we will show you how to reuse these plastic cassettes to pour your own polyacrylamide gels that can be stored for weeks, thus providing your lab with a source of inexpensive, ready-made gels for routine protein electrophoresis. Okay, let's get started.
Prepare clean Invitrogen Nupage Novex minigel cassettes. Remove about 1 teaspoon of epoxy and completely mix the solution with a pipette tip. When you have finished mixing the epoxy, apply the epoxy on the back of the front cassette where the front and back will come in contact. When you finish applying the epoxy, make sure there are no breaks in the line. Insert a sheet of paper about the thickness of the bottom opening and place the back of the cassette on top. Remove the paper, and this prevents any epoxy from entering the bottom opening. Binder clip the edges for a tight seal and let the cassette sit overnight for a full cure.